Washington State. Oops. Welcome everyone to the Washington State Kelp Research and Monitoring Workgroup. As you all know, or many of you know, this is an open forum for information sharing and collaboration. And we meet quarterly, 4 times a year, and we provide meeting agendas, notes, and recordings on the website. So if you miss anything, you can feel free to go there. And to join or leave this list, just send us an email at nearshore@dnr.wa.gov. And finally, I want to mention that we have our meeting dates, our proposed meeting dates for 2024. They are January 24th, May 15th, and October 16th. So if anybody's got some really big event that we should change them, please let us know. If not, in about a week or two, I'll send out appointments for those dates. And then finally, in the chat, could everyone please introduce themselves and provide any updates that they'd like to share and request any topics? So we're always looking to share good research that's in progress or just completed. So let us know. And with that, I'm going to pass the facilitation on to Elizabeth Spalding, who will step us through the announcements. And then following about 30 minutes of announcements, we've got 3 half-hour slots of detailed talks. One on juvenile salmon and forage fish in nearshore kelp forests. One on an update on what's going on at the Elwha, which is a fascinating project. And then finally, an update on Squaxin Island, our innermost floating kelp bed. So with that, I will stop sharing and pass it on to Elizabeth.
Great, thank you. So welcome everyone. We had a couple of announcements, so we are going to move through some lightning round updates, and I'm going to kick us off. So the first one is that I wanted to share with everyone that I have moved into a new position on the Habitat Strategic Initiative. So I am now the policy lead for the Department of Natural Resources on that team, which means we have a new marine vegetation implementation strategy lead with us today. So I am happy to introduce Bree Turner. She is joining us from the NOAA's Office of Coastal Management, and she is now on week 3. So she's just getting her feet wet, but we will be handing over the facilitation of this meeting to Bree at the next meeting. And she is going to actually share with you a little bit about our most recent investment priorities that were released last week that we think would be of particular interest to you all. So I'm going to hand it over to Bree.
Hello, and thanks for the warm welcome to this community. I've been hearing a lot of great things about you all. And I'm jumping right in at an exciting time. Just as a reminder, the Habitat Strategic Initiative Lead is a state agency team co-led by Washington Department of Fish and Wildlife and Department of Natural Resources. And this team is funded through EPA's cooperative agreement under the National Estuaries Program. And so what this team is tasked with doing, our mission is to enable and accelerate habitat protection and restoration in Puget Sound across various focus areas, including marine vegetation. And so that's the area that I'll be leading. And we do this through a variety of pathways, including strategic planning, collaborative partnerships, and funding. So here is the hook. In regard to funding, last week, very pleased to say that we have announced our 2023 funding priorities for our Habitat Initiative team. And the priority includes one of the priorities is very relevant to this team. It is new science, synthesis, and analysis for improved protection and restoration of kelp and eelgrass in Puget Sound. So this is right spot on for this community here. And right now, we've announced this investment priorities. We probably will expect an RFP to hit the street early next year. So don't have a lot more information I can provide you at this time, but I'll include some information in the chat so you can get a little bit more background on the investment priority, and happy to answer any initial questions that you have. And we look forward to ensuring this funding opportunity gets widely spread around this community and beyond. I look forward to working more with you. Thank you.
Thanks, Bree. Okay, we are just going to move quickly through our updates. So the next one we have is Jeff Whitty to talk about the Kelp Plan status update.
Thank you, Elizabeth. As mentioned, my name is Jeff Whitty, and I work with the Northwest Straits Commission. I am the coordinator for the Puget Sound Kelp Conservation and Recovery Plan, as many of you know it as the Kelp Plan. So I'm basically here to give a very quick review of kind of the progress we've made on the Kelp Plan over the last year, which does include the publication of a Kelp Plan status update. And before I begin, I just want to mention that when I talk about we, I'm not talking about just Northwest Straits Commission. I'm also talking about the Kelp Plan Advisory Committee, as well as a lot of our partners, many of you which are here, which were really helped with all the efforts of this last year. And to that collective we, I just want to say thank you. So this last year, we focused a lot of our efforts on understanding where we are with the Kelp Plan actions, to create that baseline to kind of continue off from, but to also compare to in future years. And we also gathered input on how we can continue to move forward as a community. And so this was a multi-step process. And the first step was to create that Puget Sound Kelp project inventory, which is basically a collection of information on the various projects and efforts that occurred in the Puget Sound with kelp conservation and recovery between about 2020 and 2022, and how they related to the Kelp Plan actions. And so then the second step was to take all that information and give it to the advisory committee for the Kelp Plan. And with that information and their in-depth knowledge of kelp and kelp ongoings in the Puget Sound, they then ranked, or excuse me, scored the status of the different actions within the Kelp Plan, which was a massive undertaking because there are 65 of them. And then the third step was to hold a workshop, the Kelp Plan Action Workshop, where Puget Sound Kelp partners and stakeholders assessed the advisory committee's status scores and adjusted them if need be, but also generated and ranked lessons learned and next steps for each of the actions within the Kelp Plan. And then we took all that information from all those steps and we created a summary, which we are now calling the Kelp Plan Status Update, which is now online and available. The purpose of the report was to provide a very brief overview of the advancements we have made within the, each of the goals and actions of the Kelp Plan since its publication up through that workshop time, and to report on those top ranked lessons learned and next steps generated at the workshop to help guide and inform future movement with the Kelp Plan. And for the sake of time and spoilers, I'm actually not going to go into the results of the status update, but I want to encourage you all to flip through it yourself. Hopefully it's, you know, pretty readable and easy to get through. One thing I do want to discuss though is that going through all this process, and hopefully within the report is very evident that there has been an amazing amount of effort that has been put into kelp conservation recovery within just in these last few years. However, in saying that, I do need to clearly state that we are still very early on the Kelp Plan, and it still will take years of continued support, funding, and increases in effort to get even the more advanced actions of the Kelp Plan to their intended stages. So basically, I just want to say, great work. Let's keep it up. And from here, we will continue to look at the collected information to better understand the natural sequence of the actions and where we want to emphasize our energy. So for that information, please stay tuned. I will say that you can find the status update and the Kelp Plan on the Northwest Straits Commission Kelp Plan webpage. I'll put that link in the chat. In addition, we recently updated the Kelp Plan webpage. So please let us know what you think. We will update it on an as needed basis. So we like to hear what works, what does not work. So with that, I will end and say thank you very much.
Awesome.
Oh, if you have any questions, email me. I'll put that in the chat as well.
Okay, let's keep moving then. I'm going to hand the floor to Cynthia Harbison and Max Showalter to give an update on the statewide kelp and eelgrass health and conservation plan.
Thanks, Elizabeth. Good morning, everybody. My name is Max Showalter. I'm Deputy Policy Director at DNR, and here to give you a quick overview about the kelp and eelgrass prioritization plan. So just as a quick reminder, I know many of you are already familiar, but for folks who are not, in 2022, a law was passed that tasked DNR with identifying at least 10,000 acres of kelp and eelgrass habitat for conservation and recovery. Since that time, we've been working very hard to fulfill that requirement, coordinating closely with the scientists at DNR, a panel of experts on a working group, many of whom are on the call today, working with tribes, state government, and the public to understand what we value about kelp and eelgrass that we want to be able to protect and recover with this plan. The draft plan is now out for public review, and I will throw a link into the chat and encourage you all to please take a look at the draft plan and provide your feedback. I want to emphasize that we are also looking for feedback on next steps of the plan. So what we've presented in the plan is a two-step prioritization framework that first looks at the statewide level to give us a starting point and asks where are these high value areas that we want to initiate the conversation in. But starting in 2024, we will be doing more targeted conversations at the sub-basin scale. So based on the floating kelp indicator, we've grouped out sub-basins in Washington. We added an extra sub-basin, a brand new sub-basin in Grace Harbor, and we are prioritizing 3 pilot sub-basins for targeted conversations starting in 2024. The intent for these conversations is to be able to get more localized information, deeper context, and recognition that what we have presented in the report so far is not all the information that we need in order to make decisions about how we prioritize these habitats. So starting in 2024, Cynthia and I are going to be working hard with the folks on this call, folks from DNR, to start these conversations in the South Puget Sound subbasin, the Grace Harbor subbasin, and the eastern Strait of Juan to Fuca, with the intent that we'll have conversations in all the subbasins and we'll consider areas statewide, but starting with those 3 pilot subbasins for the conversation. So again, I want to encourage you to please send us feedback through the form that I'll link in the chat. The feedback is due October 20th, so that is this Friday, but I want to emphasize that even though there's not a lot of time left to fill out the public review survey, there will be lots of opportunities in the future to engage with us and let us know what you're thinking about these subbasins and the other areas as we move forward. Thanks very much.
Great, thank you, Max. Okay, we are now going to move to Jasmine Schuster to give us an update on the BC-Washington Kelp Node.
Thank you. I'm just gonna share my screen real quick. I have a couple slides to show. Is that looking okay?
Looks great.
Yeah, hi everyone, I'm Jasmine. I'm the Kelp Node coordinator. Here to provide a couple of updates and give a brief introduction. Many faces here that I see of people who are already in the Kelp Node, but also a couple of new names and faces. First update, straight on the title slide, we have a logo, so that's exciting. Very happy about this beautiful logo. Brief intro for the kelp about the Kelp Node. So we're a network that is operating in BC and Washington waters that aims to advance kelp conservation management and recovery in transboundary waters. So really, it's about working together across the border by implementing and expanding collective actions through working groups and linked kelp science activities. And so the Kelp Node is endorsed through the UN Ocean Decade and falls under the Marine Life 2030 program. As I mentioned, the Kelp Node structure is structured around these 6 working groups that tackle shared actions. And the 6 working groups each address specific themes related to kelp. The first one tackles canopy kelp spatial dynamics. The second group is about subtidal kelp forests and kelp ecosystem services. The third group is focusing on kelp drivers of change and stressors. Fourth group is about kelp early life histories and restoration. Working group 5 tackles participative kelp science training and engagement opportunities. And then all of these 5 working groups are sort of synthesized through the data integration group, working group 6. So that brings together all the cross-cutting elements. And so where are we at now? The Kelp Node just restarted its meetings this October. We took a bit of an extended summer break during field season. So that means we're now in a third meeting cycle. At the first 2 full cycles of meetings across all the working groups, we're kind of focused on mapping out goals and priority actions. And many of these goals are aligned with the Kelp plan. We're now over 150 participants strong, which is great to see across Washington and BC, but we also have some participants from California and Alaska. And then I just wanted to give a few examples for each of the working groups for what the work that is being tackled looks like, just to give you an idea. Obviously not exhaustive, but working group 1 is working on a sensitivity analysis of remote sensing methods for regional kelp mapping. The second working group, subtitle Kelp Forest Group, is working on a methods paper that compares subtitle diving-based monitoring programs that are operating in BC and Washington. That will hopefully enable decision makers to figure out what methods work best for their specific objectives. Working group 3 is working on an evolving georeference database of kelp stressors, so aiming to provide a public resource that gives people insights for all the kelp stressor data that is being collected in BC and Washington, and what that looks like. Working group 4, which is about restoration and early life histories, is working on an open access guidelines and best practice documents for restoration. So an idea that is being discussed with that is whether we want to sort of adapt a BC Washington version of the global kelp restoration guidebook that the Kelp Forest Alliance has put out. Working group 5 will provide some training for community-based Indigenous-led monitoring. So currently talking about whether we can host a workshop in a centralized location where people can come and receive a whole variety of kelp-related training in one push. And then working group 6 is augmenting kelp data discovery and access, so developing workflows to integrate all the work that is happening across the other 5 working groups and the data and products that are coming out of it. So if you're not already in the Kelp Node, here's a scan code to register for one or all of the working groups, and I will also share a link in the chat. Thanks.
Great. Thank you, Jasmine. Okay, now we're moving into lightning talk, so even quicker updates. The first one I want to hand to is Pete Dowdy, who's going to talk about a HSIL project that is getting launched. Pete, are you here? Helen, do you know if Pete, do you want to do the update if Pete's not here?
I saw Pete's name. I wonder if he's having connection trouble, but I'll start and he can cut me off if he's not.
That sounds good.
Having connection trouble. So I'm happy to announce that a collaborative project between DNR, UW, University of Chicago, and many others, including, boy, there are just so many of us, Reef Check, PSRF, etc., is going to look at bull kelp resilience and loss at a set of 15 sites throughout Puget Sound where we have long-term monitoring data. And we will be doing detailed environmental sensor monitoring, both continuous and snapshot. We'll also be monitoring the canopies, both basic canopy extent, but also morphometrics and measures of condition. Kathy Pfister is going to be looking at kelp productivity, carbon, nitrogen, just all sorts of things about photosynthesis. And then UW has a fleet of divers, some of which are here, who are going to be looking at what's going on under the water. And so you know, really getting and looking at benthic conditions, what vegetation and also what invertebrates or other organisms are there. So that's a project that we're really excited about, and we're trying very hard to link it into other efforts. So for instance, many of you know that PSRF is putting mooring buoys out around many of their sites of interest. We will be coordinating with them. So it's deeply collaborative, and it's going a lot of field work in 2024 and 2025, and we'll be sharing it as time comes. And please contact us. I'll stand in for this if you're interested in hearing more. And I do want to mention one other collaborator, which is Reef Check. They have a great data set of dive data all over at sites all over Puget Sound, and we will kick this off by analyzing their data. So I'll pass it back to you, Elizabeth.
Okay, thank you. All right, moving along to Jody Toft with Puget Sound Restoration Fund to also share a quick update on another project funded by HSIL on strengthening restoration tools. Jody, are you there?
I am here, Elizabeth. Good to hear your voice and see your face. Can you hear me?
Yep, you sound great.
Okay, great. My first reflection is I always love seeing a full court press in action, and this continues to be a full court press on kelp research, restoration, and conservation. So, yay, team! Let's keep it up. So for my less than 2 minutes I have here, I want to highlight, and this is another complementary HSIL project to the one that Helen just talked about. And thank you, Bree, for giving the introduction to what HSIL is. So for the project that PSRF is leading, we are doing a 4-year project. Again, this is with a big coalition. So our separate recipient coalition includes, in alphabetical order, Port of Seattle, Reef Check Foundation, Samish Indian Nation, Seattle Aquarium, Squaxin Island Tribe, Suquamish Tribe, the Nature Conservancy, Washington Department of Natural Resources, and last but certainly not least, University of Wisconsin-Milwaukee. So thank you, big team, for being a part of this project, which is called Strengthening Restoration Tools for Rebuilding Kelp Beds in Puget Sound. And really the needle we're trying to thread here is how do we develop the tool that we've already been using at PSRF to rebuild kelp beds in central Puget Sound? How do we make that tool, how do we test that tool and see how well it works in different parts of the Southern Salish Sea? That tool being to grow seeded kelp line, seeded kelp line, and then deploy that underwater on the sea floor on anchored lines, and then that seed grows up into these lovely kelp curtains, which has been working really well in central Puget Sound. But how does that work when we take that technique, try it with different seed sources, which will pull before they become reproductive if they're not hyperlocal? Okay. How does that work when we try it out with maybe how does Squaxin Island seed work in central Puget Sound? What happens if you try that in the San Juans? Do we see that kelp seed that has different origins actually leads to different levels of success and failure when we're looking at kind of growth, timing of maturity, amount of reproductive material for material that we're letting go reproductive? How does that look from the ecological community point of view? How does that compare to adjacent wild beds? So kind of really taking a much more rigorous approach to understanding how this kelp that we're growing in our kelp lab, which Betsy will talk about a bit later, how does that work when we actually put it in the water, and how does it look when we compare it to wild beds? So that's what we'll be working on over the next 4 years with this coalition of partners in central Puget Sound, in South Puget Sound, up in the San Juan Islands, and in the Strait of Juan de Fuca. And you'll be hearing more about that over the next 4 years. Thanks.
Thanks, Jody. Okay, last lightning round update, Caitlin Magel and Wendell Raymond to talk about another project that identifies current and future risks to marine vegetation in Puget Sound. I know Wendell.
Thanks, Elizabeth. Hi, everyone. I'm Wendell Raymond. I'm a nearshore ecology research scientist at WDFW, and just wanted to give a quick update on a project that I'm collaborating with Caitlin Magel at the Puget Sound Institute at UW-Tacoma. So this is funded through Puget Sound Partnership scientific research funds, and we'll be getting off the ground in about a month or so here. And so the title of the grant is Distribution of Current and Future Risks of Anthropogenic and Climate Change Threats to Marine Vegetation in Puget Sound. So it's kelp and eelgrass. And the central goal of the project is to create a spatially explicit risk assessment of current and future threats to kelp and eelgrass. And a key part of that is identifying the relationship between threats, which come in many different forms, and where the habitats occur. And this work is largely inspired by a paper that I'm putting into the chat right now, which uses this methodology. I personally am not super familiar with it, but this is what the paper is based off of, or this project is based off of. And a key element of this that I will be leading is convening a group of kelp experts to rank threats to kelp. Ranking threats is a critical part of how this methodology works. And while there is a pretty good ranking of threats for eelgrass from the Rantham paper or report that many of you are probably familiar with, we would like to do something similar for kelp, with the understanding that there is much more limited data, but this will be our first shot. So some of you may be hearing from me in the coming weeks if you're interested in joining this work group to do this threat ranking. So that's all I have, and we'll definitely keep this group updated as the project moves along.
Excellent. Thank you, Wendell. All right, inexplicably, we are actually ahead of schedule, so that's excellent news. So we are going to move straight into our first full-length presentation. I'm going to hand it over to Amelia Gallagher and Ann Schaefer. They are going to talk about work that they have been doing on dynamics of juvenile salmon and forage fishes in nearshore kelp forests. And we will have some time for Q&A at the end. So feel free to put your questions in the chat if you'd like, or we can move into raised hands at the end. So I will hand it over to Amelia.
Perfect. I'll do my best to keep us ahead of schedule. Let me get my slides up here. All right, hopefully that should be shared quite nicely. So, hi everybody, I'm Amelia Gallagher. I'm an undergrad with Western Washington University, and I'm an intern of the Coastal Watershed Institute. So Dr. Ann Schaefer, who's also on the meeting today, she invited me to share this talk in her stead today, and I'm going to be discussing our findings on the dynamic interactions between kelp forests, forage fish, and salmon. And this is research led by Dr. Schaefer at the Coastal Watershed Institute, with partnerships from the University of Victoria, Western Washington University, and the University of Washington. So as we all settle in for the first full-length talk of this work group meeting, I'm going to remind everyone of some of the characteristics of kelp that are most relevant to our work specifically. As we talk about kelp, we're considering any of the large brown seaweeds in the order Laminariales. And these include both overstory and understory kelps, but I'll be focusing today on overstory forest-forming kelps, which form migratory highways along our coastlines. These are foundational where they're present because they're providing habitat, they're providing food resources, they're changing the physical environment, and they're a driver of patterns in fish and zooplankton presence. And finally, the reason why many of us are here today is kelp abundance is in flux in many coastal systems, and it's declining on a global scale, as we know. So I'm focusing today on our work on the Olympic Peninsula. This ArcGIS map from DNR shows the extensive distribution of kelp along our coastline, and you can see how it creates that migratory highway for many species. It's important to note that our study sites are specific to this region. We're in the Strait of Juan to Fuca, so more research is going to be needed for the remainder of the Salish Sea and beyond. And specifically in our region, here are the key fish we're going to be focusing on, starting with salmon, which, as you all know, are incredibly foundational to our ecosystems. If you take this map at left of all salmon-bearing rivers and streams in Washington state, many portions of the state are visually covered with these streams that salmon are using. And we have this diversity of many salmon species using our waters here in Washington. We're also looking at 3 key species of forage fish: herring, surf smelt, and sand lance. And the forage fish show their importance right there in that name. They're critical prey that many other species are foraging on. And this graph shows summary results of seining work by Frick et al. Over several years. And this shows just how dominant forage fish are as a guild. Those 3 species that I'm going to be talking about are on this graph in green, orange, and light blue. So it's really the majority of the fish that this seining effort found absolutely dominant. And forage fish are also incredibly important regionally for tribal cultures. This graph at left summarizes archaeological findings of the Tuitsin site of the Lower Elwha Klallam tribe. That's a former village site that was excavated. And there, the clupeiforms, that's herring, they were by far the most abundant guild of fish discovered at that site. And that importance continues into the modern day. They're harvested by many tribes across many species of forage fish. So our work focuses on investigating the dynamics of interactions between these 3 guilds. First off, a vital dynamic of kelp is its seasonal variability due to dimorphic life history. It's extremely sparse in winter, and then it creates these beautiful, lush forests that you've all seen in summer. Here on the left is work by Dr. Ann Schaefer and Dr. Dave Parks. The extreme left and right ends of each graph represent February, while summer is right there in the middle. So you can see how fleshy red algae died back and was replaced by an abundance of kelp there in summer in this study site, which was in Puget Sound. We're likely all familiar with the basic life history of salmon. They're born in rivers, they emerge as alevin in gravel beds, become fry, and they migrate downstream to nearshore zones to grow, rest, and feed. And the salmon will spend about 3 to 5 years offshore before returning to their natal rivers to spawn. Crucially, when the juvenile salmon emerge from these rivers into the nearshore, at first they're in inches and centimeters of water. They're not in meters of water. They're very small, very vulnerable, so they're focused on an extremely specific habitat need. And kelp beds are one of the first habitats that these young salmon encounter as they're moving into the nearshore. So forage fish have an interesting life history related to their spawning as well, but for today, we're going to emphasize just their migration and feeding. That's for brevity and because none of these forage fish have spawning behavior that uses overstory kelps. Forage fish larvae emerge from nearshore spawning zones, and just like salmon, they're using other nearshore zones, including kelp forests, to grow, rest, feed, and migrate off to their adult offshore habitat. So we know a lot about the spawning process of forage fish and of salmon, but there's a lot missing from our knowledge of their migration patterns, as well as their interactions. The forage fish and salmon are arriving to nearshore zones, and specifically kelp forests, at the same time. So there's a lot missing of our knowledge of what's happening when they're both arriving. So that's been the goal of the Coastal Watershed Institute, to understand and illustrate the dynamics of kelp forests, juvenile salmon, and forage fish, and how we as humans relate to those dynamics. We address these questions through field studies, hypothesizing that juvenile salmon and forage fish would be more abundant in kelp forests than in adjacent non-kelp zones, that zooplankton communities would be more abundant in kelp forests as well, and in particular, zooplankton that associate with kelp as a structure and that are preferred prey for these fish when they're juveniles. Also hypothesized that the use of kelp forests by these fish would differ seasonally, and that the interactions of forage fish and salmon would also differ seasonally and differ by species. So to address these questions, what we actually did is used long-term data sets for fish presence snorkel surveys and zooplankton dropnet surveys, and finally 6 years of video snorkel surveys along permanent transect lines. And these noted interactions between salmon and forage fish on the videos. So all of these studies were conducted in 2 healthy kelp beds in the south central Strait of Juan de Fuca. This is Crescent Bay and Freshwater Bay, and Freshwater Bay was featured on the cover slide for this entire work group today. It's a beautiful place. And you can see the location of both on this map at the lower left, right here in the south central Strait of Juan de Fuca. So what have we actually found? And what can I tell you today about these dynamic interactions? First, herring, salmon, and surf smelt do use kelp beds more than adjacent non-kelp areas, but sand lance don't. So taking a look at these graphs, the proportional presence of each fish in kelp beds is shown in green versus adjacent open water zones in this light blue. And we found that herring, salmon, and surf smelt were using kelp significantly more than these adjacent open water areas. Whereas for sand lance, we did not find any significant difference between the two zones. When we modeled which factors were best predictors of whether these fish were present, kelp did come up as a top predictor, again for herring, salmon, and surf smelt. Location, that's freshwater bay versus crescent bay, was a significant predictor, but only for sand lance and surf smelt. These were seen much less at freshwater bay out of the two sites. This is potentially because freshwater bay is part of the Elwha drift cell, which at the time of sampling was more degraded than today in terms of natural sediment transport. So now taking a look at zooplankton. Surprisingly, the overall zooplankton abundance, diversity, and richness were not significantly different between kelp and adjacent open water areas. That's from looking at all zooplankton together. So here, again, kelp zones are in green and adjacent open water zones are in light blue. I have diversity on the left and species richness on the right, and there was no significant difference in either metric looking at the entire community of zooplankton. However, we also actually identified these zooplankton samples down to the lowest taxon we could, and zooming into zooplankton taxa that are associated with structure, we unsurprisingly found that abundance was significantly greater in kelp zones. So here looking at decapods on the left and harpacticoid copepods on the right, their abundance was significantly greater in kelp zones. And this is unsurprising because both are known to be taxa that are associating with structure. They're literally living on the kelp. But we didn't just pull these just because they were statistically significant and because they're showing on the kelp. This is meaningful for our overall look at dynamics because these taxa are preferred prey for juvenile salmon and forage fish. They're eating both of these taxa of zooplankton when they're first arriving to kelp beds. And again, when we modeled these data, kelp came up as a top predictor of abundance for both of these two key phytal-associated zooplankton groups. So the takeaway is that general abundance of zooplankton doesn't appear to be driving salmon and forage fish to be using kelp beds. Phytal-associated zooplankton specifically, being their preferred prey, it's likely part of the story of what's bringing them there, but there's more to that habitat function. And a major piece of that is the evolving relationship between salmon and forage fish, because the salmon transition to eating fish as they grow. And to explain more of that habitat function for juvenile salmon and forage fish in kelp forests, we'll look at the results of some of this beautiful footage we got in video snorkel surveys, which was analyzed for interactions between the fish. So videos from these snorkel surveys were analyzed for fish counts, for presence of kelp, as well as the position within the kelp, being actually in the bed versus on the edge of the bed. And 4 types of interactions were categorized. So here on these diagrams, the salmon is represented in black, whereas any forage fish are represented as these outlined fish. And the interactions were categorized as adjacent, as the salmon intermingling with a school of forage fish, or 2 types of predatory behaviors. That's a salmon quickly approaching a group of forage fish, or of course, a successful predation event. And again, all of these behaviors were modeled relative to many potential predictors. So let's take a look at the actual results. This graph is showing all of the interactions that we observed throughout the study, and they're organized by month. That's May over there on the left, September on the right. So this is really highlighting the active season there in the kelp beds when the kelp is growing and the fish are using this area. So in May, we begin to see juvenile Chinook and sand lance showing up to this area. But in June and July is really when it comes alive. So many more juveniles are arriving. This includes coho, herring, and surf smelt. So we see increasing interactions. During this time, zooming in on the actual type of the interactions, we found that herring and sand lance are more important for salmon foraging than surf smelt. We actually didn't see a single instance of predation on surf smelt throughout this study. What we did see were juvenile salmon interacting with surf smelt in a different way. They were intermingling with the schools. It suggests possibly mutualistic schooling between the two species for additional safety in numbers. Then, after this wonderful, rich period of activity in June and July, we did see interactions declining in August and September as the fish are starting to move on. Lastly, we found that salmon species, position in the kelp bed, and in many cases the month of the year were significant predictors for these interactions. Looking more specifically at where these interactions happened, 78% of all interactions and 100% of predations that we observed were associated with kelp. And 60% of those kelp forest zone interactions happened specifically along the edge of kelp beds. And I want to highlight this graph at the top right. You can see all of our interactions categorized by whether kelp was present or not, kelp being in green. And specifically during June and July is when we saw the vast, vast majority of these interactions between fish taking place within the kelp during that really active, really rich period there in the kelp beds. And here's an example again of what these kelp-associated interactions can actually look like. So I'll briefly summarize our main conclusions of these studies before elaborating on the conservation implications. First, the abundance of these fish is significantly higher in kelp beds. Overall, zooplankton abundance is not driving this effect, but phytal-associated zooplankton abundance and diversity is playing a role. But the phylogenetic shift to eating fish is what's really driving the salmon to preferentially use kelp beds. Juvenile Chinook and coho predate on herring and sand lance, but not on smelt. And finally, the interactions between juvenile salmon and forage fish differ by season, and the interactions are more common in and among kelp. So just the next time you're in a kelp bed in June, and by the nature of the 100+ people in this work group today, I think that a fair few people will find themselves in a kelp bed sometime next June. Just something fascinating to think about is at that time, juvenile salmon exiting their natal rivers, forage fish migrating from their natal beaches, and kelp growing at incredible rates are all happening, all arriving at the same time in these areas. It's like a celebration of the beginning of a lifelong association. So taking a look at some of the conservation implications, we're all here because we're interested in preserving kelp. And these critically associated fish are sensitive to anthropogenic forces in the same way. Anthropogenic impacts on kelp differ worldwide, and some areas are in greater trouble than others. And specifically here on the Olympic Peninsula, our kelp beds are doing well at this moment in time. On this graph, any color other than blue represents growth. The fact that they're doing well right now emphasizes the need for conservation while they're in this state, because we know they're very sensitive to anthropogenic impacts like oil spills. Here's an example from the 1991 Tenyo Maru spill at the mouth of the Strait of Juan de Fuca. This significantly damaged kelp respiration and photosynthesis. Unsurprisingly, these same spills have terrible impacts on salmon and forage fish as well. And even worse than oil spills, because it happens on a continual basis, is stormwater. These studies at Wright show the lethal physiological effects of stormwater pollutants on juvenile salmon. And in the top right, you can even see it visually. I mean, looking at this comparison between a control, healthy larval fish versus a fish that's been exposed to stormwater pollutants, you can see the physiological differences. And of course, these same impacts are affecting forage fish as well, because they're very often spawning along heavily developed urban shorelines. This graph from WDFW shows the almost extirpated herring stock at Cherry Point. That stock is shown in red. And it also shows just the overall downward trend in herring spawning biomass over time. So your key takeaways from today are that, of course, kelp, but also salmon and forage fish are dominant features of our region that are all dynamically linked. Kelp forests provide key habitat for these fish as they migrate. This use has extreme seasonal and regional variation. We were talking in the results section about the difference between May to September. We didn't even talk about winter. There's incredible variation there. And kelp, salmon, and forage fish are physically linked to each other, as well as to larger geographic, hydrodynamic, and water quality processes. So disrupting those processes through anthropogenic impacts disrupts habitat and its key ecological functions. So restoration is an option, and when undertaken, it must focus on understanding the functional process. Why did the change in kelp forest occur? And restoration has to focus on understanding and restoring that process. Even so, restoration is often expensive and incomplete. So conservation, focusing on the natural shoreline processes in each region, is best for healthy kelp forests in the right places and their vital use by salmon and forage fish. Taking a look briefly at next steps of this research, as I said, we focused purely on the Strait of Juan to Fuca and studied 2 sites. So we're looking to expand this research to the larger Salish Sea ecosystem, and that means we get to partner with more folks. So we're building an interdisciplinary team. Partners so far include the University of Victoria, Friends of the San Juan, and hopefully others. Dr. Ann Schaefer, lead scientist at SeaWe, is here in the group today. And if anybody is interested in collaborating, she'd be the right person to reach out to. Briefly, I want to thank the very large team of SeaWe interns and staff that have contributed to this project over the years, as well as our large suites of partners and supporters that have made the work possible. And with that, I'll say thank you and ask if there's any questions.
Great, thank you, Amelia. So we do have one question in the chat that we'll start with, and then folks can either enter additional questions into the chat or they can raise their hands and we can call on them, but we do have about 10 minutes, so plenty of time. The first question is from Hugo. It says, do you know what is the shoreline designation along your research sites under the local shoreline master program?
I don't. I wonder if Anne does. She's here. My familiarity with shoreline master programs is city specific, and these are quite remote sites. So I don't know what the specific designation is, but I'm not sure they're being closely managed under a shoreline master program.
Okay, Anne, did you want to add anything?
Just first, I want to say great job, Amelia. Amelia is an undergraduate, so boy, big hand clap for her. That was really well done. No, I don't know off the top of my head what the designation is, but you can easily find that if you just get on the Clallam County webpage. It will be there. And I think their shoreline master plan, if I remember correctly, and I could be wrong, I think it's being updated now. But anyway, yeah, thank you.
Okay, we'll move to Jason. Looks like you have your hand up.
Yes, hi, great talk, Amelia. That was really well covered, and kudos to you and Anne and CWI over the years for pulling this off. More of a context question than anything else. Your last point on the last slide, we're planning to expand this work into Puget Sound next field season via kelp legislative funding. So add us to your last bullet point there. Anne already knows this because I sent her an email about it, and Helen knows it. But as part of all the kelp work happening next summer, there's a few things going on, but we think we can pull off student snorkel surveys at a number of sites around the Sound. And so, yeah, would love to be in on that, talking methods and sites. And a lot to talk about there. I think a lot of us, we all recognize there's a huge data gap for the rest of the sound, which is a large area. And there's probably going to be differences, one would think, on, juvenile salmon proximity to the source deltas they're coming out of, life history, transition to saltwater. There's going to be a lot of things we can learn. So love to gather with you all and other people for data collecting next field season.
Okay, we have a number of questions in the chat, so I'm just going to start moving through them as time allows, Amelia. The first one seems pretty quick. It's from Ian Miller. Where are the open water transects? Are they just next to the white transects shown in the map, a certain distance in depth?
Yeah, so it was essentially a double back procedure. So snorkelers would go on one transect line through the kelp and double back quite close to where the original transect was. They were set essentially just far enough so that the same region would not be double counted.
I'll add a little bit more to that, since Amelia didn't do any of those surveys. The non-kelp area was at least 10 meters away from the kelp area. We needed to stay within the same depth, but also wanted to make sure that we were off the kelp influence. Yep. Oh, and also I do want to quickly reply to Jason's comment. We're thrilled that they're getting some funding and they're actually going to be doing this in the Salish Sea, and we'll look forward to talking with him and getting a work group together and making that work as effective as possible. We've learned so much over the decades. It would be great to be able to impart that knowledge and really, support Jason and the rest of the larger group in their work. So we'll look forward to that next steps. And I guess, Jason, will you be coordinating a dialogue on that, or do you want us to do that? Just let me know.
Yeah, I'm happy to do that, Ann, and maybe you and I can touch base quick. We can think more about this in the next few months. There's no rush, really. So I think just gathering folks who are interested, and then, I would think by like February, for sure, we'd want to get a start on really planning on our end, at least, and others, what sites do we want to focus on? We have a lot drafted out, but we should all be working as a team on this and thinking of how to do it. So there's consistency, and if other folks are collecting data, just to make sure all that data fits together.
Right. And we also had, one of the key components of this are those kelp bed zooplankton trophic elements as well. And that's one that, we didn't candidly have the resources to address. We couldn't look at tummies, but I would say that's something to think about as well. But yeah, let's put that together and see if we can get that going. Thank you.
Okay, next question. Do more recent surveys of coastal kelp beds since the 2018 data you shared tell a different story? I heard anecdotal evidence in 2022 from coastal tribal elders about the disappearance of long-standing beds. And that's from Megan Chadsey.
So,
go ahead. Oh, sorry. I'm going to let Helen answer that one. The graphs that Amelia didn't include, I believe, were from as recent as, I think it was 2022. But so that I know of, the information is pretty recent. One of the areas that we're working in is the Elwha drift cell. And there's a great series of graphs from Helen's longstanding work that specifically shows the response of the overstory kelp to the dam removals. And there's been a little bit of a rebound from that event, but the bottom line is that prior to the dams coming out, the kelp distribution along the Elwha nearshore was much reduced. And when those dams went in and the sediment processes were disrupted, the shoreline hardened and the kelp distribution in the Elwha drift cell expanded dramatically. And so when that process was at least partially restored, that sediment delivery occurred in 100 years of sediment to boot, that kelp distribution changed again. So there has been some change in the Elwha drift cell. And we're not expecting it to come all the way back, but that's okay. That's the way the ecosystem is supposed to be working. And I would strongly urge everybody that's looking at kelp planning to add that as an element to your analysis factor, what the restoration actions have been along the shoreline where you've seen kelp change. I know personally at Lincoln Park, the kelp beds on the north and south of that point have changed, but I also know that the Corps of Engineers had a very large shoreline quote unquote erosion control project there that caused the shoreline to soften dramatically. And so a kelp loss there would make, logical sense and would be an ecosystem restoration. So that is what I will offer.
I'm going to jump in and say that the next talk by Melissa Foley is going to go into amazing detail on what happened at the Elwha. And so these two talks are linked. So stay tuned to hear a whole lot more about both the disappearance and the natural recovery of kelp. And then I'm going to continue in the chat talking more about areas of loss. Want to open up the main discussion.
Okay, I think we have time for one more question, so I'll read that to you, Amelia. It's how far from kelp beds were your adjacent sites, and were there any comparison sites that were far from existing kelp forests?
I think that's the same question that we discussed a little bit earlier. So the distance was 10 meters so that we could fully compare sites within kelp versus adjacent to kelp. And we were really looking to see how these fish are using kelp forests. So we did not compare to sites that didn't have kelp forests nearby, because we were interested in where these fish were actually present.
Mm-hmm. Okay, I'm going to let Helen continue to address some of those questions in the chat, and I think we might move on. Thank you very much, Amelia. Okay, we are now moving on to Melissa Foley, Steve Rubin, and Ian Miller to talk about a recent publication highlighting findings from nearshore subtidal community response during and after sediment disturbance associated with dam removal.
Great. Well, thank you everyone for the invitation to share the results of the nearshore work that's been happening alongside the Elwha dam removals and beyond. So my name is Melissa Foley. I am a science director at the San Francisco Estuary Institute, but did most of this work while I was with the U.S. Geological Survey. Steve Rubin and Ian Miller are here today too. So Steve and Ian, feel free to jump in. Yeah, and thanks also to all of the other folks who have really been pivotal in this work and who have contributed to this over the last 12 years or so. So this story that I'm going to tell today really started out as one about dam removal. But I don't want to really focus too much on dam removal itself. So, I think this story has been really well told. These two dams at the time was the largest dam removal in the world, released nearly 100 years of accumulated sediment from behind these two dams. And because this is what we were focusing on, the way that we set up the monitoring for this project was really focused around sediment and the effects of dam removal. Over time, however, that story evolved to be more than that, and it's now a story about multiple stressors that are happening in the nearshore, both at a small scale like the sediment, as well as at a regional scale. And the recovery trajectory of kelp communities in this area. So again, the expected stressor when this survey design was set up was sediment. So sediment associated with the dam removals began in really in earnest in 2012 through 2014, while the dams were being removed. In summer of 2013, another stressor emerged, a marine heat wave that lasted nearly 3 years, known as the blob, and then also a short marine heat wave in 2019, and now a new marine heat wave arriving now in 2013. Likely related to that heat wave was a third stressor, the sea star wasting syndrome, which hit in 2014. So here we have a combination of local and regional scale stressors that I think really highlight the importance of long-term data sets for understanding how kelp forest communities are responding, what types of stressors they're resilient to, and how they recover from these multiple stressors over time. So starting with sediment, because again, this was really the stressor that these surveys were set up to detect. So when the dams were removed, we saw an estimated 17 million metric tons of sediment discharged between 2011 and 2022, which are shown here on this figure. So this is sediment discharge on the y-axis, calendar day of year on the, fine sediment on the y-axis, calendar day of year on the x-axis. And then we have individual lines for 2012, which is the darkest brown, 2013, slightly lighter, 2014, this kind of olive color, and then a combination of 2015 through 2022 in this blue line here. And so what this is showing here is that in 2012 and 2013, there was an extraordinary amount of sediment that was discharged from the Elwha. In 2014, that discharge dropped by more than an order of magnitude. And the scuba surveys that I'll talk about here are just highlighted in this gray box here. And you can see as well that even during that spring and summer time period, that sediment discharge was quite elevated during the spring and summer during the dam removal period. The picture here on the right side of the screen is showing the plume, an example of a plume coming out of the Elwha River. And for the most part, due to the straits and due to the currents in the Strait of Juan de Fuca, this plume often is pushed to the east of the river. And so you'll see in our study design how we took this into account. So the second type of effect from this increased sediment coming out from the dam removal was burial. So this effect was mainly confined to the area immediately around the mouth of the river, where the effects of turbidity were also likely highest. So at the mouth, we saw about 400 meters of delta expansion between 2012 and 2016, and up to 5 meters of deposition, here shown in this middle figure, in that red area. So we actually lost some dive sites where they were formerly in about 20 or so feet of water. We could walk to them from shore, but then others highlighted here by these light gray triangles were persistently buried throughout the course of sediment being delivered into the near shore. And this distinction I'll talk about more as we get into the data, but so thinking about these site groups as buried, again, in these light gray triangles, and then sites that were unburied, and that will be a key distinction here. Along with this burial, we also saw a shift in the grain size of the substrate. So on the eastern side of the river, at these two gray triangles, and I'm hoping you can see my cursor here as I'm pointing to them, those changed from cobble to sand. And then on the western side here, these other two triangles, they changed from sand to mud. So also an important distinction there that I'll talk about later. Okay, so how are we surveying these sites? So these sites, so a core set of sites was sampled using kind of a revised method of subtidal surveys that was developed by PISCO, or the Partnership for Interdisciplinary Studies of Coastal Oceans. And this method is used along the West Coast. Reef Check has also adopted a very similar method. And essentially what we did is we had semi-permanent sites with a center post that we would find. And then there were two transects, one to the east and one to the west of 30 meters length. Along each of those transects, kelp were enumerated in a 2 meter wide swath, invertebrates in a 1 meter wide swath, and then fish in a 1 meter by 1 meter by 1 meter cube as we swam through the water column. These surveys were done on a yearly basis for the most part, mainly in August to early September. So we had 15 dive sites located near the Elwha River mouth. Again, this was mainly to capture the effects of the dam removal, as well as 2 control sites at Greenpoint, which was about 20 kilometers away from the river mouth. In this figure here, you can see that the density of sites is higher to the east of the river mouth, again, because of that sort of pushing over of the plume to the east. But we also had sites to the west, mainly in Freshwater Bay here. So again, these green circles are the 4 sites that were persistently buried, and then the blue sites are what we're calling the unburied sites. So just to give you a sense of what the kelp community looked like at these sites prior, they're mainly dominated by prostrate kelps. There are some erect understory kelps like Pterogophora and Pleurophycus. But for the most part, they're really these prostrate kelps. So what happened to these communities and how did they respond to sediment? So I'm going to go through a series of figures that look like this, so I'll just orient you first. So density as individuals per square meter shown along the y-axis here, and then time along the x. And I'll be showing data from 2011, which is our pre-removal data point, and then 2000, and then through 2022. So the control sites are in this pink color, the buried sites are in green, and the unburied sites are in blue. And what showed up very quickly for nearly every species of kelp was a very quick decline starting in 2012 and persisting through 2014. And again, this was expected as that during that main period of really high sediment delivery into the coastal zone. We don't see that similar decline at the control sites, suggesting that they were sort of outside of that sediment effect zone. But density did decrease at the control sites in 2014 and 2015, potentially as a result of the marine heat wave, which is consistent with regional patterns of reduced kelp during that time. And then things kind of separate there. We get 2 different trajectories here. So from 2015 onwards, we see that kelp rebounded very quickly at the unburied sites, while density remained more or less unchanged at the buried sites. And then at the control sites, we see some increases later on towards 2019 and into 2022. These were not significant changes, and so likely within the realm of variability of those control sites. The amount of change that we saw in the kelp community composition was as we expected, but always good to see. So this is a non-metric multi-dimensional scaling plot. And again, the colors are consistent here. Control is in this red, the buried sites in green, and the unburied sites in blue. And what an NMDS plot shows here is just points that are closer together are more similar to each other, and points farther apart are dissimilar from one another. We just have labeled 2011 and 2022 for ease of interpreting this figure, but I'll dive into each of these individually in a minute here. But just to show that the changes in community composition were greatest at the buried sites, followed by the unburied sites, again, both of which experienced effects of the sediment. And then changes were most minimal at the control site. So if we dig into this a little bit further and look at each of those individual site types. So starting with the Elwha buried, what we saw is a dramatic shift in composition away from pre-dam removal conditions in 2012, moving further away through 2014, and then somewhat moving back towards those original conditions, but not getting quite there, and kind of stabilizing starting in 2018 around a sort of static community composition at those buried sites. And this figure to the right here is just showing what species changes happened during that time. So again, 2012 through 2016 here at these sites, really nearly a complete loss of kelp due to the burial and the change in substrate. And then in 2017, we start to see some species coming back in the system, particularly Pterogophora, but still at much lower densities than they were before the effects of dam removal started. And then at the unburied sites, what we see is essentially a return to those pre-removal conditions by about 2018. So, we see this big divergence during the sediment input. And then once that turbidity is decreased towards the end of 2014 and into 2015, we see that community starts to converge very quickly back to what it looked like prior to dam removal. So two different trajectories here in the kelp system, mainly driven by that difference between buried and unburied. In those plots, you may have noticed that there was one species that did persist through what we lovingly refer to as the kelpocalypse, and that was Pterogophora. And so these are sort of what Pterogophora looks like under normal conditions. And what we saw at most sites was something that looked like this during that heavy sediment input period. So for the most part, the stalks were there, but the blades were severely damaged, likely due to abrasion of the sediment and potentially, burial and unburial at these places. It was one of the few species that was able to come back at the buried sites, but in limited numbers, and only once that substrate started to stabilize and wasn't sort of undergoing very rapid sort of burial, unburial processes. So at the unburied sites, that plot that I showed earlier really showed that kelp started to take off in 2015, but we actually started to see some signs of recovery in late 2013. So much to our surprise, when we descended onto our sites, there was actually algae, which we hadn't seen for a couple of years. And what we were seeing predominantly were recruit of red algae, but also of small kelps that were coming in as well. These don't really show up in the data because we have a size cutoff for kelp when we count them and include them in our surveys of 30 centimeters. So these weren't showing up in the 2013 and 2014 data per se, but we were starting to see this late season recruitment that was coinciding as turbidity levels dropped in the near shore. So that's the story of kelp, and we saw those losses immediately. For invertebrates, the story is a little bit different. So this is the same type of figure here as I was showing for kelp, with density on the Y, year on the X, control, buried and unburied. And what we see here is that it took longer for the invertebrate community, particularly at the buried sites, to respond to this increased flux of sediment. And that was really driven by these sites were not buried until 2014. And so that's when we see the biggest decrease at the buried sites. At the unburied sites, we didn't see we don't see that decrease in invertebrate density at those unburied sites. And it actually increased beginning in 2014. And that level stayed relatively constant through 2022. So at the buried sites in green here, we saw a loss of a lot of different species, but there were some winners that actually emerged with this change in substrate, predominantly Dungeness crabs and gooey duck clams, both of which have economic importance to the lower Elwha Clallam tribe. And again, just to kind of put these different stressors on here, so during that time of sediment, we really don't see an effect and only see an effect at the buried sites in 2014. No evidence of an effect of that marine heat wave per se on the invertebrate communities. And then in this way of looking at the data by density of all species together, we don't see a signal here of sea star wasting syndrome, but I'll get into that in just a minute about, looking at the data in a slightly different way. So the increase in invertebrate density at the unburied sites, in part, was due to increases in schizobranchia, so this feather duster worm here. And we're not completely sure of the mechanism. We do know that there was a positive correlation between the increase in these feather duster worms with fine suspended sediment load, suggesting that potential with increased particulate matter accompanying that sediment being delivered into the near shore, that there was an increased food stores for these filter feeders. It's also possible that with the kelp being wiped out, that there was less kelp abrasion for these species, and they were able to take hold and then persist even after the kelp returned. Our design was not really set up to figure out what these mechanisms were. So these are really hypotheses of why these species increased in these unburied sites. And then in terms of sea star wasting syndrome, Pycnopodia and Henricia were hit hardest at our sites. And just showing a couple of figures here on the right. Again, this is density on the y-axis, year on the x. And this black circle here on the plot to the left is showing that's essentially the loss of Pycnopodia at our unburied sites that persists actually nearly all the way through 2022 with no recovery. On the other side here, this is showing starting in 2014 and then peaking in 2015 is an increase in Mesocentrotus franciscanus, so the red urchin, following the loss of Pycnopodia at our sites. So we know these are correlated, that doesn't necessarily mean causation. And we also saw an increase in some other species, including bivalves, corresponding with the loss of sea stars at our sites. So again, we weren't really set up to be testing for an effective sea star wasting syndrome. These are, really just looking at the correlations between the loss of sea stars and the emergence of other species at a similar time frame. And then finally, changes in fish abundance. So we really only saw significant changes in fish at the sites that were buried. And here, what we saw were changes that were mainly driven by changes in the substrate type at those sites that were buried. So for flatfish, we saw an increase in abundance at the sites that changed from gravel to sand. So the sites here in blue were now present at sites where they were not before. For the poachers, which were mainly seen on the west side of the mouth, these actually decreased as that habitat changed from sand to mud. And then for snailfish, they increased when that on the west side, when that substrate changed from sand to mud. The other species that we saw major changes in were sand lance. And so these sites, we don't have densities because they're really challenging to count when we're doing our surveys, because they're just darting all over the place and in these huge schools oftentimes. And so instead what we were looking at was occurrence during the dive. And so what this figure is showing here is that on the east side, what we saw was the presence of sand lance. So we did not see them prior to that substrate changing from gravel to sand. But as soon as that did change to sand, we saw sand lance at nearly every site year combination at those two buried sites. And then on the west side, we saw a decrease as that went from sand to mud. So really driven by habitat changes for fish. So just to sum up what we saw, so kelp effects were immediate and similar across the buried and unburied sites during that time period when sediment effects were greatest. So in that 2012 to 2014 timeframe. Kelp at the unburied sites recovered quickly as soon as that turbidity decreased, presumably below a threshold that was supporting kelp. The kelp at buried sites have yet to recover to that state prior to removal. And as Ann Schaefer pointed out, it likely won't recover to what it was prior to those dams coming out because of that habitat change and the increase in sediment coming out of the river. The marine heat wave may have had regional effects on kelp density. We see a slight effect of that at the control sites in our survey, which is reflective of more of those regional effects. The invertebrate community did significantly change at all of the sites, but in slightly different ways. So habitat change was likely driving the effect at buried sites, similar to kelp. And then at the unburied sites, it's the potential of an increase in particulate matter, the loss of kelp, and the loss of sea stars at unburied sites that are driving those changes we saw there. And that the invertebrate community at both the buried and unburied sites has not returned to a similar community composition as it was prior to the start of dam removal. And then finally, fish, we saw significant changes only at the buried sites, and again, due to that habitat change and the change in the grain size at those sites. So again, just coming back to the power of long-term monitoring is, and those data being really important to understand how communities change and whether they're resilient to multiple stressor types, and how that results in significant changes to habitat and food webs, and really the mechanisms of these different stressors in those systems. So in 2012 and 2014, sediment was really high and enough to suppress kelp recruitment. By 2014, so the levels in 2012 and 2013 maybe are not relevant to thinking about other systems. But in 2014, the amount of sediment that was coming out of the river, I think, is relevant to other sediment stressors that could be happening in systems in the near shore, such as debris flows following wildfires, or landslides, or just increased sediment erosion following increasingly extreme rain events. Increasing global temperatures are likely to result in more frequent heat waves, such as the one that kind of is just coming on shore now in Washington and Oregon, which is potentially, has potential to increase the amount of marine disease outbreaks that we're seeing in these communities. So the combination of this data set around the Elwha that was really set up for dam removal, along with the regional floating canopy surveys done by DNR, and now the establishment of a reef check monitoring site in Freshwater Bay, along with the work that CWI is doing and others in this area, will be really helpful for continuing to understand how these marine communities change over time, how the multitude of stressors that they're being subjected to affect these marine communities, as well as their ability to recover. So with that, I am happy to take any questions that folks have. And
okay, I think so. We have about 5 minutes, so let's go ahead and start with the question in the chat, and then we can see if people want to raise their hands or add some additional questions. So the first one is, let's see, do your results make you think differently about how sediment management actions could minimize impacts to benthic, invert, and kelp communities?
yeah, that's a great question. And so I think, one of the things that I have thought a lot about in looking at these results is really thinking about the type of stressor that you have with sediment, whether, in sort of the persistence of it, the magnitude, and the duration, and thinking about how those overlap with the kelp life histories of the species in the region. And so, I think in this case, this was the sediment being discharged here was very persistent, very large magnitude, and very long duration. And so, I think what we saw in 2014 is potentially, again, more applicable to other places where you don't have this, major influx of sediment coming in, where particularly for annual type kelps, as long as it seems like if that turbidity is not persisting too far into the spring and overlapping with that recruitment season, we were seeing recruitment into the system. And so, I think to me, that speaks to the resilience of these systems and, sort of but there's clearly a limit to that, right? And so thinking about, again, when is that increased turbidity occurring? Is it causing scouring? Is it causing burial? And what, those are differential effects that are likely to happen in these communities. So I think that sediment management piece is really, again, what's the persistence? What's the duration? What's the magnitude? And how does that overlap?
We have a couple of comments in the chat, but no questions. So any last questions, feel free to raise your hand. Okay, so we have one in the chat. Let's see, it says, I noticed red urchins were seen in greater abundance in the face of sea star decline. Do you foresee any impacts on kelp restoration with increasing urchins?
Yeah, that's a really good question. I mean, certainly in California, where I am, that's a huge issue. And we, and I guess I don't know, we didn't see that in the data in our system, but that's a relatively local study. So I'd be interested to know if others are seeing those impacts of urchins in other places, and whether there does seem to be a release of predation with a loss of Pycnopodia in particular. Yeah.
Helen, I see you got a hand up.
Yeah, I just wanted, first of all, I think this is an amazing study. And I just wanted to underscore how fascinating it is to see that at many sites, the kelp communities return naturally to pre-removal conditions. And that's to the degree of the same species compositions at many sites. And I don't know, was that expected? Or, to me, this raises the first of a lot of research that we need to do here about natural recovery and the fidelity of specific kelp species to specific conditions. And I'm wondering if you could say anything more about that, Melissa.
Yeah, that's a great question, Helen. And, honestly, I think we didn't know what to expect after seeing just the total loss of kelp and a pretty far distance away from the mouth. And so it was really exciting to see it all come back. And I mean, it was a little bit surprising to see how much it converged on the pre-removal conditions. And, I started, thinking about, well, okay, so what is the source of the recruits that are coming into these sites? And clearly they were not affected in a way that hampered recruitment at these sites. So what are those source populations of kelp? And I think that's something, we don't know. We don't know where those recruits are coming from. I thought it was really interesting that we were getting recruitment in the late summer, which I think we were all kind of surprised by. And so, yeah, I think there's just a lot that we don't understand about how these systems recover and why they recover the way that we do. And I guess this is maybe an instance of hope of this system was able to recover and was resilient in the face of all of these different stressors.
Okay, I think for the sake of time, that will be our last question. Thank you very much, Melissa.
Thank you.
Yeah. And we are going to move on to our last presentation for the day. We're going to hand it over to Scott Steltzner with Squaxin and Betsy Peabody and Julia Ledbetter to give us an update on the Squaxin Island kelp status. So I'm not sure which of you are taking the lead.
I will take it first.
Julia's
Okay.
going to go first, and then me, and then Scott.
Sounds great.
Yes. So as Elizabeth said, my name is Julia Ledbetter. I work at DNR in the Nearshore Habitat Program. I'm going to give a brief status update on the natural bull kelp forest at Squaxin, and then I'll pass it off to Betsy and Scott. So first, a little background. The bull kelp forest at Squaxin has been relatively persistent over the last 145 years. This map shows shorelines with bull kelp in recent years in pink, and Squaxin is circled. In terms of distance from the ocean, Squaxin is the innermost bed within Puget Sound. Shorelines have lost their bull kelp are identified in blue. We've lost bull kelp from the majority of its historical extent, about an 80% decrease relative to its maximum distribution. And the current distribution creates a striking spatial pattern. The main two areas where it persists are at Squaxin, the innermost location, and then the Tacoma Narrows, which is at the entrance of South Puget Sound. So at Squaxin Island, the natural bull kelp bed has declined 97% between 2013 and 2022. In 2013, the bed area was 8.8 hectares, and by 2022, it had declined to just 0.3 hectares. The graph on the right shows the total bed area on the y-axis by year. The first big drop we observed occurred between 2014 and 2016, and then the bed area was relatively stable until 2022 when we had observed another extreme drop. In 2022 is the blue perimeter on the map on the left. In 2023, we saw some natural recovery of the bed, which is the yellow line on the map. The bed was sparse, but the bed area increased to 1.9 hectares. And on this map, the lines and points in the middle of the bed are the outplant lines and buoys that Betsy and Scott will address next. In addition to collecting bed area, we also collect minimum and maximum depth measurements along across shore transects, which is our transect map is on the left. The maximum depth of bull kelp has contracted significantly in recent years. The graph on the right shows depth on the y-axis and year on the x-axis. The purple bars show the maximum depth of kelp along the transects, and you can see that the bed is shoaled from a maximum depth of negative 4.5 meters to negative 2.5 meters. The pink bars on the top of the graph show the minimum depth along the transects, where there hasn't been big changes. The bar for 2022 is yellow because there were so few plants that we could not sample the depth along the transects, and so we took depth at every plant. One thing to note on this graph as well is that there are a lot of transects that are now recorded as zeros as kelp has disappeared from some of the alongshore transects. The small X's on the graph represent each depth measurement. And you can see that there are less X's in recent years, while in past years there were more. One major stressor of concern for bull kelp at Squaxin is elevated water temperature. Brooke Weigel's paper on the effect of temperature on the early life stages of bull kelp showed gametophyte growth and sporophyte production thermal limits at 16 and 18 degrees Celsius. Temperature sensor data from Squaxin Island showed that in 2021, which is the red box on the graph, water temperature exceeded the 16-degree threshold for a total of 618 hours across 68 days, which preceded the 2022 decrease of kelp. In 2022, which is the orange box on the graph, the water temperature exceeded the 16-degree threshold for a total of just 360 hours across 56 days, which preceded the 2023 partial recovery. We predict that the shorter duration over 16-degree threshold in 2022 may be linked to the partial recovery in 2023. And with that, that's my super brief overview, and I'll pass it off to Betsy and Scott to talk about the restorations at Squaxin.
Thank you so much, Julia. So I'm just going to talk through some points to describe what we've been doing at Squaxin Island, and then pass it off to Scott, who's going to share the results. But I want to begin by framing this within the larger context, because it was so very interesting to hear the first presentation by Amelia and Anne describing the kelp work up there, and then the LWA work, because I want to say that for this work in Squaxin, we are working in an area where kelp is not doing well, as compared to the description of kelp along the Strait of Juan de Fuca in the first presentation. And we are also working with a tribe, the Squaxin Island tribe, that wants to do everything possible to regrow and recover a kelp forest for all the ecological and cultural reasons that kelp is so important. And one of the things we're all holding on to, even though there are obvious challenges given the temperature information that Julia just shared, we're all holding on to the fact that kelp forests have persisted here off Squaxin Island for a long time, even though kelp forests have been lost throughout much of the rest of South Sound. And there was a comeback following the precipitous decline in 2022 with this year's kelp forest, wavering around that, I think 2.5 hectares or 4-acre sort of threshold. So this is challenging work. We're all trying to figure something out. How do we regrow a kelp forest in an area where there have been such dramatic declines? So with that, this is what we've been doing. In 2021, during the kelp expedition, we collected sori material at the kelp forest off Squaxin Island, and we sent that to Dr. Felipe Alberto at University of Wisconsin-Milwaukee. And he has been archiving that material in his seed bank. And I think maybe Julia's wondering why her face is still showing there. Are you wondering that, Julia? You okay? Good. Okay, so Philippe was holding that very valuable material for us, and it was so crucial that we collected it in 2021, because in 2022, there was virtually no kelp left. So moving forward to 2023, we worked with the Squaxin Island tribe and also Department of Natural Resources to sort of come up with a plan for trying to reseed that particular kelp forest to the extent that we could. So we re-imported that material from the Alberto lab, working with Department of Fish and Wildlife to get all of those import permits. And we brought that material into our kelp lab at NOAA's Manchester Research Station, where we too are establishing a seed bank, so that we have a seed bank of kelp from Puget Sound at both PSRF's kelp lab and also at the Alberto lab. And we used that material to produce gametophytes for outplant at Squaxin Island. And we needed to learn some new techniques because typically with our kelp enhancements at another location, we've collected wild sori and we've outplanted sporophytes on seeded line. In this particular case, we had gametophytes and we needed to use two different methods. There was a gravity method that Dr. Felipe Alberto has been using that he trained our staff in. And then there's also a sodium alginate binder method that Leanne Ennis up in BC has been developing, and she shared all of those techniques. So we were using two different techniques to produce seed for a Squaxin Island outplant in spring of 2023. We really wanted to get that seed out as early in the year as possible, but we also needed to work with the tribe to get all of the infrastructure in place, the ecology blocks, etc., so that we had something to put the line out on. So as a result of a combination of factors, that seed did not go out until I think March 23rd of 2023. So it was late in the season. But amazingly, both those methods of seed production worked and kelp grew at the Squaxin Island enhancement site. And it was a little bit of a nail-biting experience for all of us because there was a very long delay from the time when that seed was outplanted to when we could actually see that kelp was growing. And Scott is going to describe the incredible ROV monitoring and dive monitoring that his team has been doing to monitor what's happening with that seed outplant. But we were getting ROV images regularly from Daniel Kuntz at Squaxin Island Tribe, and we couldn't see that kelp was growing on that line. But then lo and behold, in June, late June, during an ROV survey, we could see that bull kelp was growing off those seeded lines, and both techniques had worked to regrow kelp at that particular site. There are some challenges for sure. There were a lot of kelp crabs, and I imagine that Scott will be talking about that. And I'd love to hear more from him about whether those kelp crabs were really just very dominant during the senescence period or whether they were dominant throughout that kelp growth. So that's something that we're going to need to think about. And then we also have some next steps that we're working on. So this was our first experimental outplant of seeded kelp at Squaxin Island in partnership with the tribe to do whatever we can to try to regrow kelp there amid the challenges. But we also have plans to outplant more seeded kelp in the winter of 2024 and the winter of 2025. There are some possible experiments we're going to do to develop some new techniques as well. For instance, painting kelp gametophytes using that sodium alginate binder onto rocks. And our kelp team is going to be doing some of those lab-based experiments this fall in order to prepare us for those field experiments in the winter of next year. And I just want to call out Jesse Florendo on our kelp team. They're going to be on hand and available to answer questions about those particular seed production techniques because Brian and Jesse were the ones who were masterminding all of that lab-based kelp production. And then Brian was assisting in the seed outplant. But Jesse's on hand to answer questions. Hillary Hayford on the kelp team is as well. And I just want to say thank you to the Squaxin Island tribe. Thank you to DNR for just putting our heads together and trying to do what we can. I also want to say that this particular work to try to enhance kelp off of Squaxin Island has been supported by and made possible by funding from the Washington legislature, both in the 2021-2023 legislative session and also the 2023-25 session. So we've got resources and we're happy to be working on this. And so I'm going to pass it to Scott. And then at the end, hopefully there will be time for questions and hopefully our kelp team at PSRF is going to be available to help answer them.
Thank you. Scott Seltzer from Squaxin Island Tribe, and Elizabeth, looking good on there?
Yep, you might want to
Sure.
pop it into presenter mode. There you go. Looks great.
Okay. Zoom quit. All right. I'm also may have Daniel Kuntz from the Squaxin Island Tribe Natural Resources Department. I just want to talk really quickly and give a brief update on what we did to put infrastructure out.
I think we've lost him. Oh, he's back. Scott, are you still there? I see him. I think he's just working on his screen share. So something's going on with Zoom, and that some people got kicked out. Scott didn't get kicked out. He's still here.
Maybe what I'll do is, hold on a second. I'm going to, Helen, if you're there, I'm going to send Scott's slides to you.
She's here.
I'm here. I'm standing by, Betsy.
Okay, here they come. Let me make sure they're attached. Let's get them up on the screen if we can.
Standing by.
With your beautiful waving kelp forest there, Helen, I'm glad you're standing by.
I agree with Hazel's comment of like, not now, Zoom. This was our chance to see all the beautiful results. So I do not have anything yet from Betsy. I see Scott's still on. Scott,
still
can you hear
here.
us? Can you ask Scott to unmute, or can you unmute him, Elizabeth?
He's just unmuted himself, and it seems like he's screen sharing again, so we might be back in business.
4 years of this, and we still run into it.
Yay,
How's
Scott.
this looking?
You want to pop it into presenter view, and then I think it'll, yep, looks great.
Okay, so Squaxin kelp bed, we saw how this thing has been decreasing over time and getting shallower. So the 2021 or 2022 is what we were left as this small area shown in the green circle. It was the 89 plants. Took this to tribal council and it was unanimous. They said, do something, don't just study it, watching this kelp bed go away. So talking about some of the infrastructure that went in, we decided to put in anchors that could hold the seeded lines. And what we decided to do was go off the remnant bed from the year before within this, but it's clearly within an area that always had kelp in the past. So we were able to put in 5 anchor points within here that holds about 1,000 feet of line. And within those, you can have 8 different areas for experiments as we went through. So what we first did was try to go out in February and put in augers, earth anchors like you would normally do for a buoy, and that failed. And in hindsight, that kind of makes sense because the area is super rocky, hence all the kelp within that, and we could not get the augers in there. Daniel Kuntz and the contractor did an amazing job of going out and designing, building, and placing in less than a month these ecology blocks. So basically what they are 2x6 ecology blocks with multiple anchor points within that. But when we were designing these, we're also thinking about sitting these on rocks, and with all the pressure from the lines pulling on that, it was just going to slide them across. So significantly, there are 12 pieces of rebar that come off of these about 3.5 inches in there. This whole ecology block system, worked great. These things did not move. They were easily to hook up all our equipment to it. One of the things I like about this is at least the possibility is we could move these if we wanted to go in for future work. So while we were thinking about what we wanted to do for getting out the infrastructure, how are we going to monitor this? So this was a completely unfunded project. Betsy talked to her, we're talking about future funding coming down the road, but there was nothing with this. What we had was scientific divers within the Natural Resources Department, and we had an ROV on hand. So I think the two together worked really well. The pros with, with our divers is you can get out there and do your very detailed counting in that. It's also really nice to get the big picture to see what's going on across the whole thing. When you're looking just through a lens of a camera, you're seeing a very small area within that. The complications that I'm talking about, the divers can do is that once the bull kelp starts growing, you've got stipes everywhere. The ROV had a hard time dragging its tether around within that, but the divers can get in and out. And we know all the cons of diving. It's very expensive and takes a lot of work with that. And basically your product is an Excel sheet. The ROV worked really great, especially in the beginning part of the year. Crew could run out there on any boat and they were back by lunch and uploading 4K HD video up to YouTube that hundreds of people were looking at within a day. That worked really well until later in the season again, until, all the stipes started growing. Now they can't do full surveys after that, but they could still pop in with the ROV and look at individual areas and get it out to everybody else. So the timeline that was touched on a little bit before, we tried to get these things in with the augers in February, March, failed. The crew came together and by March 21st, we had our seeded lines out. And it was mentioned before, this was at the very end of what we expected or known that was okay, but we thought we'd be all right. Went out there, started doing surveys and just saw nothing after nothing after nothing again. So by, May 9th, we were just going out and declared the thing a failure. This bottom photo was June 5th. The crew was going out just on a boat on astronomically low tide. And that's the photo that was probably one of my most satisfying times during my professional career. There was kelp growing all over the place and it was about 3 feet long. And then we did 2 more surveys on that and finished up by September 8th. So talking about these surveys, when we were out there on June 22nd and 24th, the kelp looked good, looked pretty well healthy. There was clearly predation going on kelp crabs, and it was worse closer to the ends of the anchor points, and that was where had the most amount of kelp crabs, and very few in the middle portion of that. So clearly the kelp were coming up from the anchor points and starting to move in. By our last one on September 8th, we were going to go out there to do a stipe count and then start measuring blades. By September 8th, there was not one single blade left across the entire thing, and there were very few nematocysts. Every single stipe had a kelp crab on top of it. So thousands of kelp crabs within that. Every holdfast had a kelp crab on there. There was nothing left at that point. So getting to some of the results, this is looking at the array from the top down, the black squares we're going to talk about right now. These are the ecology blocks, and there was 3 different ways of hatchery things that were going on that were applied to just those ecology blocks in there. So it's kind of inconclusive with that. The one that had the most up on the top with 46, that's just 46 individual stipes. Was also used in the center one that had a 0 in there. So there was some growth on there, unclear why it worked in some areas and not the others. This is just the lines. So on this one, we have thousands of stipes going in here and it becomes much more clear. Everywhere that the hatchery it was used on the number 1, the hatchery work had growth on every single line, and that also applied back to the ecology blocks. And what they did with the number 3 technique did not work anywhere, did not work on the lines, and did not work on the ecology blocks. Number 2 worked on both, but it was very sparse within that. So what we can kind of say is that within the parameters that we had of putting the stuff out there late, the technique number 1 had growth across the entire thing. We don't know why it took so long for the growth to take off, a month and a half after it was planted till it did, till we started seeing it again, but at least it did work and had thousands of stipes going across this whole thing. So things that we did not quantify, want to talk about is sugar kelp. The laminaria just covers the whole entire bottom of this. You can't even see the seafloor by the time that goes through. The sugar kelp, the blades overwhelmed the shallower ends of the two lines, which also had less stipes than the deeper ones. I think what would happen with that is if the bull kelp had taken off when it was supposed to, it probably would have gotten above the laminaria and gone out, because that's what we see with the natural. As we look around, as you said, the bed expanded. Laminaria is everywhere. The sugar kelp's everywhere, but there's bull kelp coming across it. Now, if you go in there and you pick up any one of these blades of the sugar kelp, you're just going to find kelp crab everywhere in there. So don't know how far out this sugar kelp went. I don't know if this is a new phenomenon because we don't have any past surveys within this area, but it will do say pull up every single blade of sugar kelp. Going to be kelp crabs under every one of those, and as far as we were swimming out and coming back. So end of that. I believe we may or may not have some time for some questions.
Yeah, we have a couple minutes. So I want to open it up. There is a conversation already occurring in the chat. Let's see, Beth has a question. Are there any observations about succession of kelp species? I have anecdotal reports of bull kelp colonizing newly cleaned mooring buoy roads and floats, and then being outcompeted by understory kelp if left uncleaned, grazed. Who on the team wants to take that question?
Or someone not on the team. My goodness, open it up. Well, maybe, Scott, we can say that in the future enhancements in 2024 and 2025, we'll certainly think about whether or not there's some clearing of rock surfaces that we want to do, or clearing of sugar kelp, and also ways to deter kelp crabs. I mean, we're just going to try to open up the conversation and develop a plan for the next couple of years so that we can learn more about whether those types of clearing techniques can help with the efficacy of this.
Yeah, I would say the first one we want to do is definitely on the shallower lines, is go out there and clear out the sugar kelp out there to make sure it does not overwhelm the top of the lines. And then that will be able to provide some of the information about a cleared rock and whether the bull kelp can start growing on it.
Helen, I see your hand.
Yeah, I just wanted to address the sugar kelp questions and discussion. We analyzed reef check data, and it's really important to note that we saw almost complete disappearance of sugar kelp in 2022, the year that the bull kelp also died back to a fraction of a hectare. So we saw a similar loss of sugar kelp and a recovery in 2023, which was a huge relief to us because we thought we'd lost 2 species of kelp. So just to note that they did occur, they were correlated. We had huge losses of both in 2022 and then a partial recovery. There is obviously kelp species compete. We all know that. So and then the other competitor there who is non-native is sargassum, and we think that's an important competitor in the shallows and seasonally. Sargassum tends to grow up earlier in the season.
So we have about 2 minutes left. Brian, are you quick? Is it a quick question?
Yeah, hopefully.
Okay.
Thanks for that. That was really cool to watch. Appreciate the sharing it. I noticed that the one seed line group that failed seemed to be from Friday Harbor. I think that's what the little bits were indicating. And I was just curious, and maybe I missed this, but was that collected from a totally different stock, or like, is there a chance that that's due to where the sori were actually from, that's why they didn't do well? Or
Great question. And I'm going to hope that Jesse Florendo can help answer this because, and maybe Tom Mumford if he's on the line as well, because that material was actually collected at Squaxin Island, but it was held at Friday Harbor Lab and then transported from Friday Harbor Lab to our kelp lab. So it became part of our seed production process. But Jesse, do you have anything else that you want to add to that?
No, yeah, that's pretty on point. And I see both Jody and Brooke jumping into chat. They're all right. It's left over from Brooke's experiment at Friday Harbor, collected from Squaxin. They were just cultured at a different rate and used in a different way. So not sure why there was a difference in performance there, but they're all Squaxin material.
Hi, Jesse. I just had a question. I guess I never really learned the fate of those gametophytes. Did you try outplanting them alongside of Philippe's stock?
Yeah, so that was what it was in Scott's presentation, that some of them went out through that gravity settling method. That's where I'm pretty sure all the gametophytes we got from you via Tom ended up.
Cool, thanks. Yeah, those were from the very diminished kelp bed in 2022, whereas it sounded like Philippe's were from 2021. So I wonder if there was any difference there from the population size. Who knows?
Okay, so I'm going to jump in. We are at time. So I do want to respect everyone's calendars. Thank you everyone for this last presentation. I do want to hand the floor to Helen for any last comments before we adjourn.
Thank you so much, everyone. It's so great to be part of our community, and please send us information about new studies or things you want to hear about or collaborate on, and we'll look forward to seeing you at our next quarterly meeting in January.
All right, thanks everyone.